Good afternoon. I think it's evening, right? Yeah, good evening. Hello. As you can see, there were many people have joined, but I'm really sorry for the delay. थोड़ा बहुत लेट हो गया मुझे join करने को, because previous class में कुछ internet issue था. So I hope you guys don't mind. Okay? Yeah. हाँ. चलो. Yes. Good evening, Jain Vichitra Durga. I hope you guys are doing well. है ना? चलो. 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 Yeah. Okay, guys, so without any further delay, let's start today's class. Okay, so as you know, my name is Ghazala Firdaus and I'm one of the biology faculty in Economics. Okay, so before starting our classes, there is a normal protocol that we take the technical check. Just let me know if we have any internet issues. कि कोई हम लोगों को इंटरनेट इश्यू है तो प्लीज बता देना. दिस इज द टेक्निकल एरर, अगर कुछ है तो उसको जानने के लिए. Okay. Okay. So as you can see, everything is clear. My voice is clear. I'm perfectly audible. Great. That's very great. Okay. So guys, we have been doing this chapter biotechnology principle and processes, and we have come to the end of this chapter. And today we are going to finish off this chapter. ठीक है, आज हम लोग इसको chapter को finish करेंगे. ठीक है. So before finishing, I'm going to ask you some basic questions. मुझे याद नहीं है कि मैंने ये last class में किया था या नहीं. अगर मैंने homework दिया था तो we will discuss this. Okay. So let's read the question. Which of the following enzyme is used in case of fungus to cause release of DNA along with other macromolecules? Okay. So this is very easy question. I'm going to launch the poll for this question. Please make sure you answer in the poll. Okay. Very easy question. We have discussed this in the last class. है ना, हम लोगों ने last class में इसके बारे में discuss किया था. Very good, very good. As you can see, people have answered, so I'm ending the poll. Okay, तो मैंने poll को end कर लिया. So share नहीं कर रही हूँ because there are only 2 people and you have answered it correctly. Okay, so let's discuss now. So which of the following enzyme is used in the case of fungus to cause the release of DNA? So this comes under the process called as isolation, है ना? Isolation of genetic material. ये हम लोग पढ़ते हैं isolation of genetic material. ठीक है, तो genetic material का जो isolation होता है, or in other words gene of interest. For this, what we have to do? First, we have to break open the cell. I had told you first we will break open the cell, and then there are cell walls. ठीक है, so cell wall होता है, cell membrane होता है. We have to break it all. We have to break it all. For the bacterial cell, what we took? For the bacterial cell, we took the lysozyme. है ना? For the plant cell membrane or the cell wall, we took the enzyme cellulase. Okay, we took the enzyme cellulase, and for the fungal cell or the fungi cell, we took the enzyme that is chitinase. Okay, इतना हम लोगों को पता है. So after this, what we are going to do? When we break open the cell membrane, there is a cytoplasm. In cytoplasm also there are many macromolecules like protein, okay, lipids, RNA. So we have to digest that with the using of different enzymes. Okay, तो ultimately हमको क्या extract करना है? हम लोग को extract करना है DNA, and that purified DNA is received or taken out by the process called as spooling. ठीक है, तो एक स्पूलिंग का प्रोसेस होता है, तो स्पूलिंग प्रोसेस करके वी हैव एक्सट्रैक्टेड द प्यूरिफाइड डीएनए. ठीक है, सो हियर यू कैन सी इन द ऑप्शंस. So lysozyme is for the bacteria, that is wrong. Okay, and cellulase is for the plant cell, that is wrong because we are searching for the fungus. Okay, so chitinase is the enzyme which is used for the fungal cell. So amylase, if you are not familiar with amylase, it is the salivary amylase. I hope you know it. ठीक है, मुझे तो लगता है आपको पता है. Okay, it is released in the buccal cavity while the mastication process is ongoing. If you eat something, you have to chew it. तो chew करने के लिए थोड़ा बहुत जो saliva आएगा, that contains the amylase enzyme. This is also not correct. Now, during the isolation of DNA, addition of which of the following causes precipitation of purified? Okay, the purified DNA तो हमको मिल रहा है, but which is the case? यहाँ पे क्या हो रहा है actually? Precipitation of purified DNA. Okay, so I'm not going to launch the poll. Just write in the chat box if you're familiar. It's very easy question, guys. Okay, तो isolation में हम लोगों ने क्या किया था? Please answer. Isolation में we done the process for taking out the purified DNA. That process is what's called as spooling. तो spooling में हमने क्या किया? We used the chilled ethanol, है ना? We used the chilled ethanol, and this chilled ethanol was put in the test tube containing that solution, जिसमें हम लोगों ने सारा material dissolve कर लिया except DNA. And then what we did when we used the chilled ethanol, what happened? The precipitation of DNA will occur and that will start to float. ठीक है, वो यहाँ पे float करने start करेगा with the help of a special spool. Okay, this is called a spool. Okay, spool. So this spool is helped in the removing of that DNA. Okay, so this is the process while isolation occurs. Okay, the chilled ethanol is the correct answer. So if you are asking me about the ribonuclease enzyme, ठीक है, ribonuclease enzyme, ठीक है, ये किसके लिए वर्क होता है बेटा? Ribonuclease enzyme help in the degrading of RNA. Okay, proteases are done for the protein, so to digest the protein. So DNA polymerase is very simple. If you are remember the PCR technique which we were studying, है ना, तो वहां पे मैंने आपको term use किया था polymerization. ठीक है, तो same polymerization is helped by this enzyme DNA polymerase, okay, which we are going to study it the continuity. Okay, now, so we were studying the amplification of gene of interest. देखो first हम लोगों ने क्या कर लिया? We have isolated the gene of interest, and then what we did? We have cut it down that big gene of interest into the smaller pieces. Now we cut it down into the smaller pieces by using the enzyme restriction endonuclease that when cuts the DNA fragment at the specific sites, we call that as recognition sites, जिसमें 6 base pairs होते हैं. ठीक है, तो recognition site में cut कर लिया and then we got the small DNA fragments. So these DNA fragments are done what? इनको हम क्या करेंगे बेटा? We are doing the gel electrophoresis to separate the DNA fragments. So how we are going to separate that? We are using the electric current. जब हम current use करेंगे, then what happen? Near the agarose gel, जहाँ पे pits and wells हैं, उसको हम cathode लेते हैं और उसका opposite side anode. Okay, when we turn on the current, what happen? The smaller size the DNA fragments, they move very farther away. बहुत दूर तक जाएंगे and the heavy ones will go very short distance. They travel very short distance. So this is how we separate the DNA fragments. And after this process, we have obtained the DNA fragments. तो इसको हम क्या करेंगे? We use the stain ethidium bromide. वो stain use करके हम लोग elution करेंगे. Elution means what? Removing of that gene of interest from the agarose chain. After the process of elution, what we got? We got the what? Gene of interest. Now, with only one gene of interest, we cannot do the work. I have told you we have to keep the backup. That is why we have to multiply the number of gene of interest or amplify the number. So for the amplification purpose, we are using a specialized process. We call it as PCR. Okay, PCR is what? Polymerase chain reaction. Okay, so this was introduced by the person called as Kevin Lewis. Okay, तो इस PCR में हम लोगों ने देखा था 3 major steps थे, है ना? If you remember, I'm just going to wrap it up, मतलब थोड़ा revision करके, ठीक है? So first step was denaturation. So what is denaturation? देखो हम लोग इनके पास क्या था बेटा? We had the spiral double helical circular DNA. and we had this type of DNA. So first what we did, we straightened it down. Okay, first हम लोगों ने इसको खींच के straight कर लिया. It looks like ladder now. So after that we have to break the hydrogen bonds. Okay, we did the breaking of hydrogen bonds between the two strands. अभी for example यहाँ पे A T C G, okay, T A ऐसा अगर कुछ है तो यहाँ पे हम लोग क्या करेंगे, T A G C A T. तो इसको हम क्या करेंगे, बीच वाले हाइड्रोजन बॉन्ड्स को ब्रेक करके we will separate the two DNA strands. We will separate the two DNA strands. Now, these two DNA strands we call them as template strand. Okay, we call them as template strand. That is nothing but the parent strand. So while doing this process, we are maintaining the temperature at 92 degrees Celsius. ठीक है, हम लोगों ने टेम्परेचर कितने में मेंटेन किया, 92 degrees Celsius. Now, after that we are going to do a process called as annealing. Okay, annealing में क्या होता है, I have told you the word meaning itself is adding or joining, adding or joining. Now what we have the template strand or also called as parent strand. इसको हम क्या बोलते हैं, parent strand. Now, so template strands are created. Now we will use the primers. है ना, हम लोग क्या use करेंगे बेटा, we will use the primer. Okay, I have told you in the last classes raw materials. So raw materials को जो जो चाहिए था उसमें primer भी added था. So primer is the short, a DNA with the single stranded. Okay, short DNA sequence with single stranded condition. तो वो primer को हम लोगों ने यहाँ पे add कर दिया. ठीक है, primer को add कर. So always what happen, primer attaches only at 3' end. देखो DNA में 3' end है, तो ये primer जाके 3' end में ही attach. ठीक है, I hope you remember the DNA replication. So whatever the replication process happens, same thing happens here, but वहाँ पे ना replication में natural process है, and this PCR is done in the artificial laboratory, मतलब in vitro condition में हो रहा है. ठीक है, चलो. So here primer attaches at the 3' end. So polymerization will start to happen. So what is polymerization? If you want to understand what is polymerization, देखो बेटा, amino acid, amino acid, amino acid, these are the monomer. Okay, these are the monomer units. Okay, if you join these amino acid together, it will become a protein. ये protein बन जाएगा. So protein is a polymer. Okay, amino acid is a monomer. Similarly, here monomers are nucleotides. Okay, nucleotides. I hope you remember the nucleotides. ठीक है, बायोमोलिक्यूल्स याद होगा. एक शुगर होता है, एक फॉस्फेट ग्रुप होता है, और होता है एक नाइट्रोजनस बेस. ठीक है, ये सब मिलके एक न्यूक्लियोटाइड होता है. When we continuously add the nucleotides here, then what happen? One by one polymerization will occur, and we get the ultimately strand of DNA. So ultimately, हमको एक strand of DNA will be obtained. Okay. So that process is called as polymerization. So polymerization starts by adding the nucleotides. So in this condition, temperature is maintained at 55 degrees Celsius. Temperature कितने में maintain किया है? 55 degree Celsius. ठीक है? तो यहाँ पे polymerization हो रहा है, new nucleotides are getting added. The same thing happened in the opposite strand. Okay, primer attaches at the 3' end, and the nucleotides are started to join. Okay, and always remember, this polymerization, it happens in the particular direction. Which is that direction? It happens in the okay, 5' end to the 3' end. ठीक है, so यहाँ पे 5' है, यहाँ पे 3' है, तो इसी डायरेक्शन में पॉलीमराइजेशन होगा. So this is annealing. Okay, adding of primer and adding of nucleotide. Now the last stage is called as extension. So extension में तो बहुत सिंपल है बेटा. यहाँ पे हमारा स्ट्रैंड था, यहाँ पे प्राइमर था, तो अभी ये प्राइमर ऐड करते करते एक नया सा डीएनए का स्ट्रैंड ही बना देगा. ठीक है, there is a addition of new nucleotides and it will create the DNA strand. Okay, so this is extension. Okay, so it will join, and ultimately we will get the new DNA. So what we understood in the extension process, we have got the टू कंप्लीट सेट, ठीक है। इतना हो जाने के बाद यहाँ पे जो है वो हाइड्रोजन बॉन्ड्स क्रिएट हो जाएंगी एंड वी गेट द 2 DNAs, ओके। हाइड्रोजन बॉन्ड क्रिएट हो जाएंगे एंड वी गेट द 2 DNAs. सो हियर 2 complete set of DNA is obtained, okay. 2 complete set of DNA strands is obtained. So here temperature is maintained at 72 degrees Celsius. Confirm कर लेते हैं, 72 degrees Celsius. Okay, 72 degrees Celsius में यहाँ पे temperature maintain हुआ है. And what we got here, see, we started from one, okay, one strand. तो इससे हमको क्या मिल गया? 1 plus 2. So this is what PCR is. Okay, so this you can say there is a drastic multiplication of DNA strand. Okay, so similarly, this is a cycle. If you want many gene of interest, then what happen? You will do the many cycles. मतलब एक cycle में 2 आएंगे, 2 cycle में फिर 4 आएंगे, 4 cycle में फिर ऐसे 8, 16. So they will keep on coming. Okay, so 2, 4, 8, 16. So this number keeps on increasing. So PCR reaction runs under the cycle system. So from one cycle we get the 2. Okay. So this is how extension happens and ultimately we get the 2 DNA strands. So in this process you can see this is the PCR machine. Okay. So this is the PCR machine and this is the DNA sample. We have taken primers, we have taken the nucleotides, then Taq polymerase, very important. Why? To maintain this functioning, we want the thermostable polymerase, and this thermostable polymerase is obtained from the bacterium aquaticus, Thermus aquaticus. ओके, तो यहाँ पे बफर चाहिए क्योंकि इन सबको मिक्स करने के लिए एक बेस या फिर एक बफर, देन देयर इज अ PCR tool. Okay, so first what we did the denaturation and we maintained the temperature at 95 degrees Celsius. So 92 to 95 degrees Celsius is maintained. Okay, तो यहाँ पे क्या होंगे? Strands will break. Okay, the strands will break down and then what happen? There is a process of annealing. So annealing में क्या होगा? Primers यहाँ पे आके add करेंगे and new nucleotides will go on adding. You are seeing no new nucleotides will go on adding. So this is annealing. But here temperature is maintained at 55 degrees Celsius. Again in the third step, we are getting the complete synthesized new strand. Then the temperature is maintained at 72 degrees Celsius. So from one DNA strand, we got, okay, 1 plus 1, we got 2 DNA strands. So this is how amplification of gene of interest is done by the method of PCR. Okay, so now, so what we were doing here, देखो हम लोग क्या क्या कर लिया? हम लोगों ने isolation कर लिया, cutting कर लिया, amplification कर लिया. Now we want to insert. What we want to insert? This gene of interest, what we have produced in the larger amount. ठीक है, larger amounts में जो हम लोगों ने produce किया है, इसको हम क्या करेंगे? What we are going to do it? We are going to insert it in the plasmid. Okay, so there is a gene of interest and we insert that gene of interest in the plasmid. Then we get the recombinant DNA. See, this is plasmid, this is gene of interest. Then we get the recombinant DNA. So this recombinant plasmid or recombinant DNA is introduced into the host. इसको हम क्या करेंगे? We will introduce in the host. Then we will allow the host to grow. देखो, our host cell is E. coli. It's a bacterium. It will multiply in every 20 minutes. ठीक है, every 20 minutes के अंदर हम लोगों को एक नया generation मिल जाएगा. Okay, so with the new formation of cells, we are doing what? We are cloning. Okay, recombinant DNA जो हमको मिला है, it will start to clone. Okay, from 1 to 4, then again like this. तो हम लोगों को बहुत सारे मिल जाए. ठीक है, so now, so when the gene of interest get incorporated in the host cell, then what happens? Our host cell will start to produce the product. Okay, when there is a gene, okay, see, you might have heard beta central dogma of life. The gene or DNA is encoding the information. वो information क्या होता है? Transcription will occur, then translation will happen, then we get the ultimate protein. So that protein is our product. So similarly here, by using the gene of interest, okay, that might be anything, antibiotic resistance gene or any kind of insulin producing gene, तो कुछ भी हो, So that is produced, okay, by getting incorporated. Chain of interest will get incorporated in the host cell and we get the product. Okay. So product मिल जाने के बाद we have to do the processing. Okay. So the product what we got is a raw material. And मतलब एक raw product है, we cannot directly sell it in the market. We have to separate it, purify it. If there is a need of adding preservatives, we have to add it. Okay. देन उसको क्या करना है, एक लेबल लगाना है, एक ब्रांड नेम लगाना है, इतना सब करने के बाद देन यू विल टेक इट इनटू द मार्केट. चलो अभी इंसुलिन को हम फिल करेंगे. So like this, these are all the methods. Now we have to see the fourth point. Okay. Now, so here you can see the ligated DNA. So ligated DNA is nothing but जो recombinant DNA है, ठीक है, जो recombinant DNA है इसको हम host cell के अंदर introduce करेंगे. Okay, when we introduce this ligated, so this recombinant DNA is called as ligated DNA. Okay, उसको ही ligated DNA बोल रहे हैं. Ligated means what? Here ligase enzyme has acted for joining the plasmid with gene of interest. Okay, gene of interest को plasmid के साथ attach करने के लिए ligase की help पड़ी थी, that enzyme. That is why they have used ligated DNA. Okay, so this is introduced into the host cell. Okay, then what happened? This process is called as transformation. I think Manjula madam has told you this. Okay, that process is called as transformation. ठीक है, तो ट्रांसफॉर्मेशन होगा तो ये कोई भी मेथड से हो सकता है. Either you can make the cell competent. Okay, how we make the cell competent? By treating it with the calcium chloride, then giving the hot और क्या कहते हैं heat shock treatment. So heat shock treatment देंगे तो ये competent कर रहे हैं host को. Other than that, we can use the gene gun method. Okay, so gene gun method use करो या फिर micro injection use करो. By any method, they are introducing the ligated DNA into the host cell. Okay, then now. So this transformation, after doing the transformation, I hope you remember, okay, I think Manjula madam has told you this. I think so. तो बस मैं थोड़ा आपको एक रिवीजन दे देती हूँ उसका. Okay, what she has told you, beta. There is a plasmid, okay, and there is a gene of interest, okay, and there is a gene of interest. So gene of interest and plasmid together, we call it as a recombinant plasmid or recombinant DNA, recombinant plasmid, recombinant DNA or ligated DNA. So anything you can use it, ligated DNA. Now this is introduced into the host cell and now what happened? Host cell के अंदर introduce करने के बाद we are allowing it to cloning, okay, multiply, so cloning. So this process is called as transformation. तो इस process को हम लोगों ने बोल दिया था transformation. Now, so after transformation, there are three situations or three cases. तीन cases हो सकता है. क्या हो सकता है बेटा? The first case, I will say the first case. So in the upcoming bacterial cell, okay, so एक हमारे पास host cell था, उसमें हम लोगों ने rDNA को डाला and then we allowed it to cloning. So further the next generation bacteria might not have any plasmid. Okay, कई बार क्या होता है, there is no plasmid entered. Okay, this is one case. There is a second case in a bacteria, plasmid has entered, but there is no gene of interest. Okay, plasmid has entered, but no gene of interest. Okay, this is a second case, and there might be a third case. Here, what happens inside the bacteria, we have the gene of interest with us. Okay, plasmid with gene of interest. So these are the three cases. After the transformation, we might get three cases. So for this, we gave the fancy name. So in सारे processes को हमने fancy name दिया है. The first condition where there is no plasmid, we call it as non-transformant. Okay, we call it as non-transformant. And for the second one, we call it as non-recombinant transformant. देखो transformation तो हुआ है. There is a plasmid present in it. But in that plasmid, there is no gene of interest. That is why no recombination has occurred. Okay, recombinant नहीं है, recombinant DNA नहीं है. So non-recombinant transformant. Okay, transformant. So this is on second type. Okay, so third type is what? Recombinant transformant. So now what we have to do, we don't want the first one and second one. We don't want first one type of bacteria and second one type of bacteria. हम लोगों को नहीं चाहिए, ये भी नहीं चाहिए. Then what we want? We want this. We want this recombinant to grow more and more. ताकि जब बैक्टीरिया में जीन होगा, then we get the desired product. ठीक है? So that gene will produce the desired product, the recombinant product in it. तो उससे हम लोग क्या करेंगे, जो इंसुलिन हमने एग्जांपल इंसुलिन का लिया था, तो दैट इंसुलिन वी विल सेपरेट प्यूरिफाइड एंड यूज इट. ठीक है, चलो. सो ये आप लोगों ने लिख लिया, फिर ये शीट नहीं मिलेगा आप लोगों को होम इसमें नोट्स में. ठीक है, चलो. नाउ so that's what is given here. Okay, that's what is given here. See, we want two things. One, we want the recombinant DNA in our host cell, and that recombinant DNA should be a transformator. These two things you have to remember. So what they're saying, if the recombinant DNA bearing the ampicillin resistant gene is transferred into E. coli, then what happens? Nodi, gene ಇದೆ ಅಂದ್ರೆ, oh my god, why I'm saying that? देखो, अगर जीन है तो फिर क्या होगा, एम्पिसिलिन रेजिस्टेंट तो बन ही जाएगा हमारा, है ना. So our cells will become ultimately ampicillin resistant because there is the gene present in the E. coli. It might be not the natural, इसके पास originally नहीं था, but हमने उसको डाल दिया तो वहां पे क्या हुआ, ampicillin resistant quality आ गई. ठीक है, so this is one of the type. And then if the transformed cells are spread on the agar plate containing ampicillin, then what happens? Only the transformants will go. Okay, so those transformants which have recombinant DNA. So this you have studied, right? जो वहाँ पे बोला था selectable marker. I think Manjula madam has told you about the selectable marker. एक tetracycline. There is a plasmid. So tetracycline, ampicillin. So in tetracycline at the BAM H1, you cut it down. Okay, then what happens? The tetracycline activity is, halted, or you have to stop. Then ampicillin active रहेगा. So ये सब आप लोगों ने पढ़ लिया है. I don't want to repeat it. Okay, so just what you have to know, we have the recombinant DNA and the transformant bacteria. तो ऐसा बैक्टीरिया को हमको ग्रो करना है in the abundant. ठीक है, so that is then after searching it. Okay, सब हो गया. After searching those recombinant transformants, we have to understand something. What is that? देखो बेटा, हमारा ultimate goal क्या है? Our aim is, okay, to produce the desirable product. Okay, so that desirable product or protein is insulin in our example. Okay, that is insulin in our example. Okay, so here in the second sentence, what they have given is very simple. E. coli के अंदर ampicillin resistant gene तो नहीं था, है ना? It was not there. So how it came? When we add it. Okay, so we have added the gene of interest. That gene of interest has the ability for resistance of ampicillin. तब ये हम इसको E. coli में डाल देंगे, sorry, E. coli को डाल देंगे, तो E. coli has the ability now. Okay, now E. coli has the ability for ampicillin resistant gene. So ये कैसे हुआ? देखो, जब जीन है तो अल्टीमेटली उसका प्रोटीन भी बनेगा, है ना? So we got the recombinant protein. So don't take the problem here. यहाँ पे टेंशन लेने की कोई बात नहीं है. Let me repeat it once again. Okay. So what happened? So we have introduced the foreign gene which has the protein encoding information. ठीक है, वो कौन सा प्रोटीन एनकोडिंग इंफॉर्मेशन है? इंसुलिन. ठीक है, सो वो जीन हमने क्या किया? होस्ट में डाला. तो अल्टीमेटली होस्ट का ओरिजिनल प्रोटीन नहीं है. ओरिजिनल प्रोटीन होता तो हम लोग यहाँ पे हेटेरोलॉगस होस्ट यूज नहीं करते. ठीक है, सो होस्ट naturally does not produce this insulin protein or recombinant protein. It is producing because you have inserted the gene of interest inside the host. That is why it has attained the ability. Now host can produce the protein. That protein is the recombinant protein. देखो, recombinant DNA से हमको क्या मिला? recombinant protein मिला. ये ओरिजिनली इकोलाई में नहीं था. आफ्टर द इंट्रोडक्शन ऑफ जीन ऑफ इंटरेस्ट हम लोगों को we got the recombinant protein. So this protein is nothing but our desired insulin. Okay, chalo. Now, so furthermore, you want to see here, cells with foreign genes can be grown in laboratory. That is true. Okay, we are using the laboratory techniques to grow the cultures. Okay, so cultures are used to extract the desired protein and purify it by using the separation techniques. See, once we got the gene of interest, we amplified by using PCR. So PCR में amplify करके बहुत सारे gene of interest बना लिए. फिर gene of interest को plasmid में डाला और transformation किया. तो transformation करने के बाद we got the transformants, non-recombinant transformant, recombinant transformant. तो first two को हमने eliminate कर दिया by the use of selectable markers and we kept only the recombinant transformant. ठीक है? So by taking this recombinant transformant, you can see that with the help of gene of interest, those bacteria have produced the product. Okay, that product is the recombinant protein. In our example, we have taken the insulin. ठीक है? तो अभी ये हमको मिल गया, ये तो छोटे छोटे अमाउंट में हो रहा है. अब हम लोगों को क्या करना है? इतना सब मेहनत करने के बाद सिर्फ एक दो बंदे को इंसुलिन मिल रहा है, तो व्हाट इज द यूज? We want insulin to produce in the larger quantity. बहुत बड़े बड़े quantity में we want to produce. So for this we are using a special vessels called as bioreactors. Okay, we are using the bioreactors. So what are bioreactors? These are big vessels. ठीक है, ये बड़े बड़े vessels हैं जिसमें हम क्या करेंगे? कल्चर करेंगे हमारे इकोलाई को. कल्चर मीन्स we are growing the E. coli. So which type of E. coli we are growing in the larger amount? Our recombinant transformant. Okay, recombinant transformant which we selected, that we have to, okay, that we have to culture here. ठीक है, तो बड़ा सा कल्चर में एक वेसल में we are culturing the E. coli. So this E. coli cell will ultimately give us what? It will give us the larger amounts of insulin. ठीक है, तो अब ये कैसे करेंगे? You have to study more about the bioreactor. ठीक है, चलो. So these bioreactors are vessels in which raw materials are biologically converted into specific product. So what is the product that we want? We want insulin. Okay, insulin is our product. तो इसको लेने के लिए हमने कौन सा raw material use किया? We used not any chemical. We used the live organism. Okay, if you remember the definition of biotechnology, what it says? Okay, it is the technique where live organism is used to make the desirable protein or product for the useful of human. Human को useful है इसलिए हम ये बना रहे हैं. Okay, by using any microbe or plant cell or animal cell or human cell. Okay, insulin production आपके next chapter में आएगा. We will discuss there in detail. Okay, कुछ spleen के cell निकालने हैं, islet of Langerhans के cell निकाल के हम अच्छे से okay, चलो अभी. So bioreactors are used to produce the larger quantity of products. देखो इतने से बीकर में क्या होगा भाई? कुछ नहीं होगा. हमारे लिए बड़े बड़े reactors. So those bioreactors can withhold the hundreds to thousand liters of culture. Okay, thousands of liters का culture हम यहाँ पे करेंगे. Okay, so for the culture of that E. coli which has the recombinant DNA, for that to grow, we want to give the optimum condition. Optimum condition का मतलब क्या है? देखो, optimal means favorable. We have to give the favorable condition. मतलब E. coli को nutrient देना है, अच्छा सा temperature देना है, optimum temperature, optimum pH, okay, optimum substrates, सब we have to provide. Then only what happens? The E. coli will grow and we will get the desired product of insulin. Okay, so now we have to study more about the bioreactor. So how bioreactor looks, how actually it works. So all these things you have to study here. ये डरना ही जाना, बहुत easily मैं explain करूँगी. ठीक है, चलो. So here With the white chalk piece I have written, this is a big vessel. Okay, this is a big vessel, and this vessel is a bioreactor. Now, inside this bioreactor, first, if you want to culture the E. coli, you have to give the food. I know the food is nothing but the nutrients like carbon source, nitrogen source, potassium, manganese, okay, sulfur, कुछ भी हो, these minerals and the nutrients that you have to provide. Okay, so these nutrients are provided by a medium. We call it as nutrient medium. Okay, we normally call it as nutrient medium. So this nutrient medium might be solid. It might be solid or it might be liquid. Okay, let me tell you about the solid agar medium. Okay, so solid is done with the help of agar. Remember that agarose shell, electrophoresis we used kiya tha. So that agar which is extracted from the seaweed or geranium or algae, तो उसको हम लोगों ने help किया. And then what happened? We cooked that in the water and that agar melted. जब melt हो गया, then what we did? We took the sterilized Petri plate and we poured that agar solution. When we poured the agar solution and we left it for you know solidifying. So cold होते ही solidify हो गया, and now in the Petri plate we have the agar medium in it. Okay, so agar medium. So after the agar medium, what happened? You will introduce the culture. So that E. coli which you want to grow, okay, that E. coli which you want to grow, you add it here, and eventually there will be the abundant E. coli colonies. Okay, the blue color which I have written, these are the E. coli colonies. This is how we grow the E. coli cell in the solid medium. Okay, but this can happen in the laboratories. Okay, it is very favorable for labs. But when we are making the culture to grow the E. coli in the larger bioreactors, the best nutrient medium is the liquid medium. ठीक है, liquid medium को हम कहते हैं liquid broth medium. Okay, broth means nothing but the liquid particles. Okay, so here liquid broth medium में what we are doing, we are taking a beaker. Okay, मतलब छोटा सा आपको बता रही हूँ. In the beaker, what we did, we have taken all the nutrients in the form of liquid. That means what, we have added everything except agar. Okay, agar we didn't use. We used all the nutrients, all the materials, and then what we did, we added the culture, our E. coli culture, and then we allowed it to grow in the optimum conditions. मतलब अच्छा सा temperature pH सब दे के हम लोगों ने allow किया grow करने को. Now in this liquid medium, our culture is growing very happy. Okay, so this is how liquid medium broth we have to imply in the bioreactor. Okay, so this is what we are implying in the bioreactor. So what happened in the bioreactor then? In the bioreactor, if you see, first you have to add the nutrients, the nutrient medium. Then what you do, you are going to add the inoculum. So आप अभी मुझे पूछोगे मैम inoculum क्या है? So the inoculum is very simple. This is the sample. Okay, sample E. coli. Okay, which needs to be cultured, which needs to be grown or cultured. Okay, so that E. coli which is has the recombinant DNA, उस DNA को हम लोग कहते हैं inoculum. मतलब ये होता है कि इसकी वजह से ही ग्रो हो रहा है कल्चर सारा. ठीक है, so this inoculum को हम लोगों ने ऐड कर लिया. So अभी जो ग्रीन ग्रीन यहाँ पे दिखा रहे हैं, these are the colonies. Okay, तो यहाँ पे जो sorry. So this green and blue is nothing but the E. coli culture. Now, we have provided everything. But after this, we have to monitor. Can I let you go? Sorry. Now what happened? We have allowed the bioreactor to work. Okay, we have everything. But there are some optimal conditions maintained. So for those optimal conditions to be maintained, we want the controlling. Okay, we want the controlling unit. You can see this is the controlling unit. In the controlling unit, you can see the temperature, pH and everything. can see the temperature, मतलब temperature ज़्यादा है या कम है, that you can see. Sorry, actually I have a dry cough for many days. I have took the prescription from the doctors, but it's not working. I don't know why. Even though I am Omicron negative, I have done the RT-PCR. I'm negative, but it is just a dry cough because I'm allergic to chalk fish. Okay, I am taking offline class. I'm situated in Haveri, Karnataka. So we take offline classes. So I'm consistently exposed to the dust, chalk dust. Because of that, I'm allergic. Okay, no problem. We'll continue. Right. So what we have done, we have to maintain the temperature. So CU may controlling unit के अंदर there is a temperature knob. If you want to increase the temperature or decrease the temperature, what you can do, there is a presence of water. Okay, ये water jacket जो है, it helps in the entry of water. For example, यहाँ पे टेम्परेचर सेंस करके कंट्रोलिंग यूनिट में बताया कि ओहो देयर इज वेरी हॉट कंडीशन, यू हैव टू कूल डाउन द बायोरिएक्टर. देन व्हाट यू विल डू, in this water jacket you send the cold water or chilled water. When you pass the chilled water, the bioreactor will get cold. Okay, that is one way. If you want to heat the bioreactor, then you can use the hot water. Okay, so by this water jacket we maintain the temperature of the bioreactor. Okay, now if you want to monitor the pH, then what happens? You can continuously add any acid or base here. Okay. For example, in this bioreactor, you want to increase the acidity. Okay, you want to make it acidic. Then what happens? You are going to add the hydrochloric acid or any other acid, okay, which makes the acidic condition that you add. Either if you want to create it the alkaline condition, you add the NaOH. Okay, NaOH is a basic solution. So that you will add, and then what happens? The solution will become, the culture medium will become the basic. So similarly, this pH pipe help us for the passing of acid or basic for maintaining the pH of culture medium. Okay, so pH maintain कर लिया, temperature maintain कर लिया. Then हमको क्या चाहिए? As our E. coli is aerobic bacteria, we want to provide the good amount of air. So how we are providing it? We are providing it by the bottom pipe. Here there is an air pipe. Okay, so this air we are not sending the normal air, we are sending the sterilized air. If you ask me what is sterilized air, what you're seeing in the nature, मतलब यहाँ पे जो दिख रहा है आपको, ये air में ना there are many airborne diseases and there are many other microorganisms. What we have to do, we have to clear all these microorganisms and we have to send the pure air. Okay, similarly, same thing is happening here. We are sending the sterile air by excluding or removing all the unnecessary waste and dust particles that are suspended in the air. Okay, so सिर्फ हमने sterile air भेजा. Now what happens? This sterile air will make the culture very aerated. ठीक है, गैस आ गया, एयर आ गया, तो जो भी ऑक्सीजन की नीड थी वो फुलफिल होगी. ठीक है, now only the 3 by 4th of the part is filled in the bioreactor by the culture medium, but the 1 by 4th part of the bioreactor is as it is. मतलब ये ऐसे ही है, ये ऐसे ही है. So now here what happens? There is a air present. Okay, that air needs to be sterilized. So for that sterilized conditions, we consistently send the steam. Okay, steam हम लोग भेजते रहते हैं ताकि यहाँ पे sterilization maintain रहे. See, ultimately we want the E. coli cell, because from the E. coli only we get the recombinant protein, that ultimate protein, that is insulin. If there is a contamination, E. coli के पेस में चलो कोई streptococcus आ गया, staphylococcus आ गया, we don't want all those bacteria to come, salmonella theme. We don't want that. We want the contamination-free bioreactor culture. So that is why we are maintaining the sterile conditions, okay, to prevent the contamination. So इतना सब देने के बाद, after providing all these things, we still notice that there is a problem in the growth of E. coli. Why? Because some bacteria which are near to the water jacket, they are getting the good temperature. Okay, मतलब optimum temperature बहुत अच्छे से मिल रहा है, and they are growing very beautifully. But the bacteria which are growing on the surface, they are not growing very beautifully. Okay, so यहाँ पे problem क्या हो रहा था? यहाँ पे कुछ एक जगह पे अच्छा grow हो रहा था, कुछ कुछ जगह पे अच्छा grow नहीं हो रहा था. So this problem was, overcome by the use of impeller. So if you ask me, ma'am, what is impeller? Impeller is nothing but a shaker. Shaker means what? Not the name Shekhar. Shekhar. कोई आपका friend Shekhar है तो उसका नहीं बोल रहे बेटा shaker. Shaking. Okay. So impeller is nothing but a rod with the blades. Okay, you can see here these are the blades. So these blades, just like a fan, there is a central rod and there are blades. So those blades, when they turn, you get the air. Okay, you get the coolness. So similarly, यहाँ पे जब वो shake करेगा, then all the culture medium will be shaken and all the necessary conditions are equalized. ठीक है, जो हम लोग optimum conditions maintain करने की कोशिश कर रहे थे, वो equal हो जाएगा. So in the top culture medium will be same as the bottom. So there is complete equality is maintained in the culture medium. Okay, so this problem was overcame by the impeller. Now, so this impeller rotates with the help of a motor. Okay, तो मोटर में यहाँ पे देखो रीडिंग होती है, तो ये जानने के लिए कि कितनी स्पीड में impeller घूम रहा है. Okay, at what speed the impeller is rotating. So all the things are monitored. Okay, now so इतना सब बोलने के बाद सिर्फ एक ही चीज लेफ्ट ओवर हुई है, that is in the open area there is also a blade present. Okay, there are also blade present in that 1/4 of the air place. So this is present here to control the foam that is produced. Okay, whenever you shake something, okay, have you seen the milkshake, strawberry milkshake? तो जब ज्यादा उसको शेक करोगे ना तो there is a formation of bubbles. Okay, so the foam or the bubbles. So we don't want that foam to occur. So that is why this plates or you can say this blades help in the breaking those foam. Okay, so foam to break them. So this is the entire structure of the bioreactor which you need to know. Okay, so chalo, in the next slide we can see. So this bioreactor is same as in the NCERT book. Okay, देखो यहाँ पे एक vessel है. This is the big vessel. Okay, and there is a culture broth that is in the liquid medium. Okay, उसके साथ कौन grow हो रहा है? हमारा E. coli cell grow हो रहा है. ठीक है, यहाँ पे देखो impeller है. There is a impeller, which is a flat bladed impeller. And देखो यहाँ पे there is a foam breaker. And we have a motor which runs the impeller. And there is a steam passing for sterilization. That passage is there. Sterile air is there. And then pH को maintain करने के लिए acido base add करने के लिए एक छोटा सा pipe. Okay, so this is the bioreactor. Now, there are two types of bioreactors. Okay, so two types of bioreactors. So first type of bioreactor is very, simple. The name itself has simple stirred tank bioreactor. Okay, stirred tank bioreactor. Okay, so here what actually happens is very simple. Okay, तो यहाँ पे क्या होगा बेटा? There is a impeller. कि एक तो bioreactor है. It is a tank. A tank because what? एक बड़ा vessel है. That's why we are using the term tank. Okay, so with the help of impeller, it is very easy to rotate the impeller. ठीक है, मोटर है तो impeller rotate करेगा. तो impeller जब rotate होगा तो stir होगा. Okay, stir means what? Shaking. Okay, have you heard stirring process? Okay, अगर आपने कहीं देखा है ना कोई कुकिंग शो में तो क्या बोलेंगे? First you have to keep the कढ़ाई, then add oil, then add onion, then you have to keep stirring until it turns brown. तो ऐसे कुछ रेसिपीज होते हैं. तो stirring means nothing but shaking. Okay, तो shaking. Okay, so stirring. So simple stir करके हम लोगों ने बायोरिएक्टर बना लिया है. That is this bioreactor. Now, there's another type of bioreactor. We call it as sparged stirred tank bioreactor. Okay, this is another type of bioreactor. So here what happened? Instead of impeller, we are using a different kind of rod-like material. This rod also rotates. This rod also rotates. It also has one type of blade here. ओके, बट ये क्या करेगा बेटा, सिर्फ एक बार रोटेट करेगा, बस थोड़ा ऐसे से रोटेट करता रहेगा. बट व्हाट हैपन हियर, द मेन डिफरेंस इज, here sterile air is sent through the pipe, but here what happened, we are sending the air in the form of bubbles. We are sending the air in the form of bubbles, and these air bubbles are moving here. Okay, so because of these air bubbles, what happened, the continuous aeration is maintained. So sparged simply means bubbles, because parched means bubble. So this is another type of bioreactor. So you have to remember these two types of bioreactor which is given in the NCERT. Okay. So now let's move ahead. Now, so I wanted to explain you one more thing. When we say fermentation, so what is fermentation? It is the process of conversion of glucose into either lactic acid or ethyl alcohol or any type of alcohol. Okay, which doesn't need the oxygen. This is a normal fermentation process. But in the biotechnology, we use the fermentation process for something that is manufacturing in a bigger quantity. Okay, bigger quantity means ज़्यादा amount ನಲ್ಲಿ ಏನಾದ್ರು ನಾವು produce ಮಾಡ್ತಿದೀವಿ ಅಂದ್ರೆ ಅದಕ್ಕೆ ನಮಗೆ fermentation process ಅಂತ ಹೇಳ್ತೀವಿ. ನೋಡಿ, fermentation anaerobic condition ಅದು different. But ಇಲ್ಲಿ ನಾವು bigger amounts ನಲ್ಲಿ produce ಮಾಡ್ತೀವಲ್ವಾ, so this is also called as fermentation process. Okay, so it is used as a common term now. So there are two types of fermentation in the bioreactor. ಎರಡು ತರದ fermentation ಇದೆ. First one is called as batch fermentation ಅಥವಾ closed type, closed system. ನೀವು ಹೇಳ್ತೀರಾ ನಮ್ಗೆ ಏನಿದು ಗೊತ್ತಾಗಿಲ್ಲ, ಹೇಳ್ತೇನೆ ನೋಡಿ. ಇದ್ರಲ್ಲಿ ಏನಾಗುತ್ತೆ, ನಾನು ಬ್ಯಾಚ್ ನಲ್ಲಿ ಆಡ್ ಮಾಡ್ತೀನಿ. ಒಂದು ಬ್ಯಾಚ್ ಹಾಕಿದ್ರೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಹಾಕಿದೆ, ಕಲ್ಚರ್ ಹಾಕಿದೆ, ಅಂಡ್ ಐ ಅಲೋಡ್ ಫಾರ್ ದ ಈಕ್ವಲೈಟ್ ಟು ಗ್ರೋ. ಅಂಡ್ ದೆನ್ ವಾಟ್ ಐ ಡೂ, ಐ ವಿಲ್ ರಿಮೂವ್ ಒನ್ಸ್ ಐ ವಿಲ್ ಆಡ್ ಅಂಡ್ ಒನ್ಸ್ ಐ ವಿಲ್ ರಿಮೂವ್. ಸೊ ಅದರಿಂದ ನಾನು ಸೆಪರೇಟ್ ಮಾಡ್ತಿರ್ತೀನಿ. ನಮ್ದು ಈಕ್ವಲೈ ಏನು ಗ್ರೋ ಆಗಿದೆ, ಏನು ಪ್ರಾಡಕ್ಟ್ ಬಂದಿದೆ, ಅದನ್ನ ಸೆಪರೇಟ್ ಮಾಡಿ ನಾನು ಫರ್ದರ್ ಪ್ರಾಸೆಸ್ ಮಾಡಿ ಮಾರ್ಕೆಟಿಂಗ್ ಸೆಲ್ ಮಾಡ್ತೀನಿ. ದಿಸ್ ಇಸ್ ಬ್ಯಾಚ್ ಫರ್ಮೆಂಟೇಷನ್. ಅಂದ್ರೆ ಕ್ಲೋಸ್ ಸಿಸ್ಟಮ್ ನಲ್ಲಿ ಹಾಕಿದ್ರೆ ಒಂದ್ಸತಿ ಹಾಕೋದು, ಒಂದ್ಸತಿ ತೆಗೆಯೋದು, ಮುಗಿಯಿತು ಕತೆ ಆಯ್ತಾ. ಆಮೇಲೆ ಬ್ಯಾಚ್ ನೀವು ಹಾಕ್ತಾ ಇರ್ಬೇಕು. ಸೆಕೆಂಡ್ ಟೈಪ್ ಇಸ್ ಕಂಟಿನ್ಯೂಸ್ ಕಲ್ಚರ್ ಮೀಡಿಯಂ. ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, ಇದು ಓಪನ್ ಸಿಸ್ಟಮ್. ಅಂದ್ರೆ ಏನು, ಇದು ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ರನ್ ಆಗ್ತಾನೆ ಇರುತ್ತೆ. ಇಟ್ ಇಸ್ ಕಂಟಿನ್ಯೂಸ್ಲಿ ರನ್ನಿಂಗ್. ದಿಸ್ ಬಯೋರಿಯಾಕ್ಟರ್ ರನ್ ಆಗ್ತಾ ಇದೆ. ನೀವು ಫ್ರೆಶ್ ಮೀಡಿಯಂ ಆಡ್ ಮಾಡ್ತಾ ಇರ್ತೀರಾ, ಯಾವ್ದು ಯೂಸ್ ಆಗಿದೆ ಅದನ್ನ ತೆಗಿತಾ ಇರ್ತೀರಾ, ಆಡ್ ಮಾಡ್ತೀರಾ, ತೆಗಿತಾ ಇರ್ತೀರಾ. ಇದು ಏನ್ ನೀವು ತೆಗಿತೀರಲ್ಲ, ಅದರಲ್ಲಿ ನಮ್ಗೆ ಬೇಕಾಗಿರುವಂತಹ ಡಿಸೈರ್ಡ್ ಪ್ರಾಡಕ್ಟ್ ಇದೆ, ದಟ್ ಇಸ್ ಇನ್ಸುಲಿನ್. ಓಕೆ, ದಟ್ ಇಸ್ ಇನ್ಸುಲಿನ್. ಸೊ ಫ್ರೆಶ್ ಮೀಡಿಯಂ ಆಡ್ ಮಾಡ್ತೀರಾ, ರಿಮೂವ್ ಮಾಡ್ತೀರಾ, ದಿಸ್ ಕಾಲ್ಡ್ as continuous culture system. ಸೊ ಇದರಿಂದ ನಮಗೆ ಏನ್ ಬರುತ್ತೆ, we get the product. So in our case we have taken the example of insulin. ಇದೇ ಇಲ್ಲಿ ಬರ್ತಿದ್ದು ಇಲ್ಲಿ ಇತ್ತು ಅಲ್ವಾ, ನಾನು ಹೇಳಿರ್ಲಿಲ್ಲ. ಸೊ ದಟ್ಸ್ ವಾಟ್ ಇಸ್ ಗಿವನ್ ಹಿಯರ್. The cells can be multiplied ಅಲ್ವಾ. ನಮ್ಗೆ ಏನು, ನಮ್ಗೆ ರೀಕಾಂಬಿನೆಂಟ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್ ತಗೊಂಡಿದೀವಿ, ಇದನ್ನ ನಾವು ಮಲ್ಟಿಪ್ಲೈ ಮಾಡ್ತಿದೀವಿ in the continuous culture system. So first what we are doing, we are draining out to the one side. ಅಂದ್ರೆ ಬಯೋರಿಯಾಕ್ಟರ್ ಗೆ ಒಂದು ಟ್ಯಾಪ್ ಇದೆ ಅಪ್ಪ, ಒಂದು ನಲ್ಲಿ ಇದೆ. ಓಕೆ, ನಲ್ಲಿ ಯಾವ್ರಿಗೆ ಟ್ಯಾಪ್. ಓಕೆ, ಸೊ ಅದರಿಂದ ಏನ್ ಮಾಡ್ತೀವಿ, ಒಂದು ಏನ್ ಯೂಸ್ ಆಗಿದೆ ಅದನ್ನ ರಿಮೂವ್ ಮಾಡ್ತಾ ಇರ್ತೀವಿ. ಆಮೇಲೆ ಒಂದು ಪೈಪ್ ಇರುತ್ತೆ, ಅಲ್ಲಿಂದ ಆಡ್ ಮಾಡ್ತಾ ಇರ್ತೀವಿ ಫ್ರೆಶ್ ಮೀಡಿಯಂ ಲೈಕ್ ದಿಸ್. The fresh medium added and removed the drained from other side. ಸೊ ಇದರಿಂದ ಏನಾಗುತ್ತೆ, ಏನು ನಾವು ಮೇಂಟೈನ್ ಮಾಡ್ತಾ ಇದ್ದೇವಲ್ವಾ ಬಯೋರಿಯಾಕ್ಟರ್ ನಲ್ಲಿ, ಅದು ಕಂಟಿನ್ಯೂಸ್ಲಿ ಆಗಿ ರನ್ ಆಗ್ತಾ ಇದೆ. ನಮಗೆ ಲಾರ್ಜರ್ ಬಯೋಮಾಸ್ ನಲ್ಲಿ we can extract the product. Okay, so ultimately we will get the desired protein, that is insulin. Okay, so this is what I wanted to tell you. Now ಇಷ್ಟು ಆಯ್ತಲ್ವಾ. ಈಗ ನೋಡಿ, ಫರ್ಮೆಂಟೇಷನ್ ಪ್ರಾಸೆಸ್ ಅಂದ್ರೆ ಏನು, ಇದು ಎಲ್ಲ ಆಗ್ತಾ ಇದೆ ಮೇಡಮ್ ನೀವು ಕೇಳಿದ್ರೆ, ಇದು ಬಯೋರಿಯಾಕ್ಟರ್ ಒಳಗಡೆ ಆಗ್ತಾ ಇದೆ. ಎಲ್ಲ ಆಗ್ತಾ ಇದೆ, ಬಯೋರಿಯಾಕ್ಟರ್ ಒಳಗಡೆ ಆಗ್ತಾ ಇದೆ. ಐ ನೋ ಟೈಮ್ ಹ್ಯಾಸ್ ಡನ್, ಬಟ್ ಜಸ್ಟ್ ಗಿವ್ ಮಿ 2 minutes. ಓಕೆ, ಬಯೋರಿಯಾಕ್ಟರ್ ನಲ್ಲಿ ಆಗ್ತದೆ. ಸೊ ಇದರಲ್ಲಿ ಅಪ್ ಸ್ಟ್ರೀಮಿಂಗ್ ಪ್ರಾಸೆಸ್ ಅಂದ್ರೆ ಏನು, ಇಲ್ಲಿ ನಾನು ರಾ ಮೆಟೀರಿಯಲ್ಸ್ ತಕೊಂಡಿದ್ದೀನಿ. ಸೊ ವಾಟ್ ಡು ಮೀನ್ ಬೈ ರಾ ಮೆಟೀರಿಯಲ್ಸ್, ನೋಡಿ ನಾನು ಪ್ಲಾಸ್ಮಿಡ್ ತಕೊಂಡೆ, ಸೆಲ್ ತಕೊಂಡೆ, ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಶನ್ ಮಾಡಿದೆ. ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಶನ್ ಮಾಡಿದ್ಮೇಲೆ ರೀಕಾಂಬಿನೆಂಟ್ DNA ಬಂತು. ದೆನ್ ಐ ಹ್ಯಾವ್ ಗಾಟ್ ದ ಇನಾಕ್ಯುಲಮ್. ಇದೆಲ್ಲ ಇದ್ರ ಒಳಗಡೆ ಆಡ್ ಮಾಡಿದೆ. ಸೊ ಆಡ್ ಮಾಡಿ ಕಲ್ಚರ್ ಮಾಡಿದೆ. ಸೊ ಕಲ್ಚರ್ ಪ್ರಾಸೆಸ್ ಆಗ್ತಾ ಹೋಯ್ತು. ದೆನ್ ಐ ಗಾಟ್ ದ ಡಿಸೈರ್ಡ್ ಪ್ರಾಡಕ್ಟ್. ಸೊ ಈ ಪ್ರಾಡಕ್ಟ್ ನಾನು ಡೈರೆಕ್ಟ್ಲಿ ಮಾರ್ಕೆಟ್ ಅಲ್ಲಿ ಸೆಲ್ ಮಾಡಕ್ ಆಗಲ್ಲ, ಇದನ್ನ ನಾನು ಪ್ರಾಸೆಸ್ ಮಾಡಬೇಕು. ಸೊ ದಿಸ್ ಪ್ರಾಸೆಸ್ ಇಸ್ ಕಾಲ್ಡ್ as ಡೌನ್ ಸ್ಟ್ರೀಮಿಂಗ್ ಪ್ರಾಸೆಸ್. ಅಂದ್ರೆ ಈ ಸೆಲ್ಲಿನ ಒಳಗಡೆ ನಮಗೆ ಬೇಕಾಗಿರುವಂತಹ ಪ್ರಾಡಕ್ಟ್ ಇದೆ. ಫಸ್ಟ್ ವಿ ಹ್ಯಾವ್ ಟು ಸೆಪರೇಟ್ ಇಟ್, ಪ್ಯೂರಿಫೈ ಇಟ್, ಅಂಡ್ ದೆನ್ ಇಫ್ ದೇರ್ ಇಸ್ ಅ ನೀಡ್ ಆಫ್ adding preservative, I will add it, and then I get the product. Now it is ready to sell in the market. Market ಅಲ್ಲಿ ಸೆಲ್ ಮಾಡಕ್ ಈಗ ರೆಡಿ ಇದೆ. So ಇದರಲ್ಲಿ ಯಾವ process involve ಆಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ that I will tell you now. Okay, so first biostream, sorry, downstream process andre it is a series of process where separation ಆಗುತ್ತೆ, purification ಆಗುತ್ತೆ after the culture stage. ಯಾವಾಗ ನಮಗೆ ಕಲ್ಚರ್ ಆಗಿ ಪ್ರಾಡಕ್ಟ್ ಬರುತ್ತೆ ಆ ಸ್ಟೇಜ್. ಸೊ ಇದಕ್ಕೆ ನಾವು ಸೂಟಬಲ್ ಪ್ರಿಸರ್ವೇಟಿವ್ ಹಾಕ್ತೀವಿ, ಕ್ಲಿನಿಕಲ್ ಟ್ರಯಲ್ಸ್ ಮಾಡ್ತೀವಿ. ಇನ್ಸುಲಿನ್ ಬಂದ ತಕ್ಷಣ ನಾವು ತಗೊಳ್ಳಿ ಮಾರ್ಕೆಟ್ ಅಲ್ಲಿ ಮಾರಿ ಅಂತ ಬಿಡೋದಿಲ್ಲ. ಆಯ್ತಾ, ಫಸ್ಟ್ ವಿಲ್ ಟ್ರಯಲ್ ಆನ್ ದ rat or rabbit or any human. ಸೊ ಟ್ರಯಲ್ ಮಾಡಿ ಕ್ವಾಲಿಟಿ ಟೆಸ್ಟ್ ಮಾಡಿ ದೆನ್ ವಿ ವಿಲ್ ರಿಲೀಸ್ ಇಟ್ ಇನ್ ದ ಮಾರ್ಕೆಟ್. ಸೊ ಫಸ್ಟ್ ಏನ್ ಮಾಡ್ತೀವಿ, ಸೆಪರೇಟ್ ಮಾಡ್ತೀವಿ. Okay, first what we are going to do, separating, and then purification is done, and then adding the preservative if there is any clinical trial ಮಾಡ್ತೀವಿ, quality testing ಮಾಡ್ತೀವಿ. ಮತ್ತೆ ಈ ಡೌನ್ ಸ್ಟ್ರೀಮಿಂಗ್ ಪ್ರಾಸೆಸ್ ಇದೆಲ್ಲ ಇದು ಸೆಪರೇಟ್ ಆಗುತ್ತೆ, ವೇರಿ ಆಗುತ್ತೆ ಫ್ರಮ್ ಪ್ರಾಡಕ್ಟ್ ಟು ಪ್ರಾಡಕ್ಟ್. ಇನ್ಸುಲಿನ್ಗೆ ಒಂಥರ ಆದ್ರೆ ಯಾವ್ದೋ ಒಂದು ಬೇರೆ ಅಮಾಕ್ಸಿಸಿಲಿನ್ ಆಂಟಿಬಯೋಟಿಕ್ ಡ್ರಗ್ ಗೆ ಬೇರೆ ತರ. Okay, so there are different types. So this is all about the bioreactors and the biotechnology. We have finished the chapter. This is your homework question. I don't have the time to answer it. Homework question idu. Answer ಮಾಡಿ ನಾನು notes ನಲ್ಲಿ ಕಳಿಸ್ತೇನೆ. Okay, so if there is any doubt, please make sure to ask it. Okay, so before that I'm launching the NPS. ನೀವು ಹೋಗೋಕ್ಕಿಂತ ಮುಂಚೆ please ನಿಮ್ಮ feedback ಕೊಡಿ. Okay, you might understand. ಮೇಡಮ್ ಹಿಂದಿ ಹೇಳ್ತಾ ಹೇಗೆ ಕನ್ನಡ ಸ್ವಿಚ್ ಆದ್ರು, ಬಿಕಾಸ್ ನಾನು ನೋಡ್ದೆ ಅಪ್ಪ ಪಾರ್ಟಿಸಿಪೆಂಟ್ಸ್ ಬೇರೆಯವ್ರು ಹೋಗ್ತಿದ್ದಾರೆ, ಬರೀ JNV ಚಿತ್ರದುರ್ಗ ಅಂತ ಇತ್ತು. ಅಂಡ್ ಚಿತ್ರದುರ್ಗ ಇಸ್ ಅವರ್ ಪ್ಲೇಸ್, ಸೊ ಐ ಥಾಟ್ ವೈ ನಾಟ್ ಟು ಯೂಸ್ ದ ಟ್ರೆಡಿಷನಲ್ ಲ್ಯಾಂಗ್ವೇಜ್. Okay, chalo. Please give your valuable feedback. Okay, and I know I'm turning late. You can leave the class after giving the feedback. Your feedback counts a lot. Okay. I have launched the poll. Please give your valuable feedback. Okay, chalo. Thank you. Bye. If you have any doubts, you can ask me. No problem. Okay, if you have any doubts, you can ask me, or else you can leave the class. So this is your homework question. I will be sending this in the notes. Okay, ನಾನು ಇದು notes ನಲ್ಲೇ ಕಳಿಸ್ತೀನಿ. Okay. Okay.